So today I thought we'd do something a little bit different and talk about the evolution of the eye. So we're going to look at the past, present and the future. So it's always been a topic that I've found personally quite fascinating over the years. So I've been looking forward to doing something like this for quite a while now. And I really enjoyed putting it together, to be honest. I quite enjoyed researching it. So hopefully you enjoy it too. So the reason I wanted to do this was just to get an understanding of how an eye can actually evolve, how we ended up with the eyes that we've got today, and we'll have a look at some examples throughout the animal kingdom that demonstrate the different stages of the evolutionary process and different types of eyes. And we're going to have a look at the present, so what's actually happening with our eyes at the moment, what kind of evolutionary changes are taking place, and then what kind of potential changes could there be in the future. So, you know, how, in terms of how we manage eye care, in terms of how our eyes are going to adapt and change, what could the future potentially look like as well. So to start off with this, so you'll all know that guy on the left, so that's Charles Darwin. So he came up with the original theory of evolution in 1859 with his book On the Origin of Species. So the premise was obviously that natural selection is the mechanism of evolution. And the basic premise is that as there are sort of mutations and changes within the genes as part of the reproduction process, slight changes that give organisms survival advantages or slight advantages over their competitors then get passed on through the gene pool, etc. But it's not the only type of method that we can have evolution from, so we'll touch on that in a second as well. But like I say, basically it states that organisms that are more adapted to their environment, they're more likely to pass on the genes that aided their success. So this, like I say, it's not a random process in terms of things just randomly changes. It's changes within genes and random changes at a gene level that force this evolutionary change. It's not, things don't just sort of miraculously appear and then change. It's these subtle changes over a long period of time. And all it takes is one generation for a slight change to happen. So as those genes are passed on, the copies are sometimes made imperfect. There might be a slight imperfection. If you imagine if you're photocopying something and there's a hair on the scanner, that hair gets passed on onto the next copy, etc. So it can always be slight little changes, slight little imperfections that are passed on, but sometimes these imperfections or these little random changes provide some kind of advantageous element for reproduction, for survival, etc. So one example, you might have two birds of the same species. One might have a slightly more hooked beak than the other. That slight hook to the beak might enable them to access a food source that their other competitors can't get to. And then from having that slight advantage, they're more likely to reproduce. And then the genes that develop that slight hook on the beak are going to be passed on to the next generation, and so on and so forth. And as we have these incremental changes over times, over generations, we'll get these more defined features that evolve as advantageously. But there's also a lot of myths around evolution, and there's a lot of misunderstandings. So first of all, that picture that we see of the ape going into the human, that picture now it was quite a common thing that people saw. It's quite a well-used example of the evolutionary process, but it's not really an accurate portrayal of what happens during the evolution. So it's kind of gone by the wayside now. But the first myth that is commonly sort of propagated with evolution is it's survival of the fittest, and that doesn't really do it justice. So the fittest don't always survive. Although a lot of the time throughout the natural selection process, things that do give an advantage are more likely to give you that chance to survive and to reproduce, but there are a lot of different factors that can contribute to whether a species survives. So although you may be the fittest, you may have the best chance of survival, if there's economic changes, climate changes, geography changes, changes further down the food chain, for example, then that's always going to have a knock-on effect. Good example, think about something like a saber-toothed tiger. Very dominant creature, perfectly well adapted to be the apex predator, but changes further down the food chain meant that it died out. So it's things like that. So the fittest don't always survive. Natural selection is not the only means of evolution either. So there's something called genetic drift, which is basically how genes are passed on through generation to generation. And these can be predominantly through sort of geographic locations. So we tend to find that genes within a certain population will get passed on more regularly because of the sort of confinements of that geographic location, for example. And in the sort of modern world, we're not as influenced by nature, particularly now as us as a species, as humans. So the whole sort of survival advantage doesn't necessarily have much of an influence on us today, because we can overcome a lot of the barriers to survival. We've got healthcare, we've adapted the environment to us, to shape to what our needs are. So we're less at the whim of natural selection than we would have been, thousands and thousands of years ago. But nowadays it is more of a genetic drift that's going to influence the evolution of us as a species. So common question is, if we evolved from apes, why are there still apes? Well, we didn't evolve from apes, we evolved alongside apes. So we share our common ancestor, our closest cousin, which is the chimpanzee, and we split from a common ancestor about 6 million years ago, which was called Australopithecus, which was just called southern ape, basically. And from those divergent points, that's where the two species evolved separately, but we didn't evolve from apes, we evolved alongside them. So in terms of why they're still apes, obviously apes are well adapted to their environments and they don't need to become more complex to survive. So it's not a case of only the fittest survive, only the most evolved survive. They're perfectly well adapted to their environment and they can survive and continue to reproduce in that sense. So species randomly form. So as I mentioned before, species don't just randomly form, they change over cumulative change, they diverge due to cumulative changes. And one of the biggest influences on this, again, could be something like geography. So, for example, you could have a population of a certain species in a certain area. There could be a flood that could cause that population to split. They could be split by a river, for example. One part of that group now have to adapt to a slightly different environment, different climate, different food sources, and so on and so forth. So you get these kind of divergence of species, and that's generally where the species tend to split. But there's different categories of species, and each species belongs to a family and a genus and a type, etc. So I'll go through that in a second as well. But generally speaking, so if you have two animals from the same species, then what defines a species is that those animals can reproduce and produce fertile offspring. However, animals that are from different species, although they might be from the same order or the same genus, they might be able to produce offspring, but that offspring won't be fertile. So a good example is a ligra, so a lion and a tiger, or a mule. So if you get a donkey and a horse, breed them together, you get a mule, but that mule is sterile, it can't reproduce. You can't then make two mules to get more mules, because they are slightly different species. So this is known as taxonomy, and this gives us the kind of the path that the different species follow within their broader groups. So we have a species, our species is Homo sapiens, and there was quite a few different types of subspecies within that genus, within that Homo genus. So obviously, got quite a long list of them there. Heidelbergensis, that's nothing to do with Heidelberg engineering. Most of them are named after different areas where they were discovered and things like that, such as Neanderthals. Rudolfensis, that's because it had a red nose. It didn't really, that was a joke. So there's different kinds of subspecies within that Homo sapiens species. So does anyone know what Homo sapiens actually means? So it just means wise man. There you go. So, which was what separated us from the rest of the hominid species. Okay. So we also have the genus. So the species all belong to a genus, and the genus is Homo. And the Homos belong to the family called the Hominidae, which then belong to the order the primates. So primates, I mean the Hominidae family include things like the great apes, so chimpanzees, gorillas. Primates include things like monkeys, gibbons, lemurs, bush babies, and then the class is Mammalia. So that those primates, the order of the primates, then falls under the category of mammals. Mammals are defined as animals that give birth to live young, so don't lay eggs, and feed milk, produce milk. Okay. So if we look at the sort of the timeline of evolution, so obviously we're talking about going from something very, very basic, something very sort of single cell, to extremely complex organisms. And just to get an idea of how long that timeline is and how long that process took. So it all starts with the sun. So the sun was approximately 5 billion years ago when the sun formed. And then when the earth formed, we started to have simple organisms around about 4 billion years ago. And then we get through this period where we come up to what was known as the Cambrian explosion. Now, this was about 540 million years ago. And this saw the biggest period of change throughout the evolutionary process. So the term explosion is a little bit sort of misleading, because this was like a approximately 20 million year period. But during that 20 million years, there was such a divergence in life forms. Still aquatic, still sea-bound, but there was so many changes and so many evolutionary processes and much more competition at that time that it was, in terms of creating new species, it was a massive explosion during that time. Then we keep going along and we come to mammals. So mammals first appeared around about 200 to 250 million years ago, with primates coming in around about 55 million years ago. And then our first sort of homo, first sort of bipedal hominid, which was Ardipithecus ramidus, that was around about 4.4 to 5.8 million years ago. And then finally Homo sapiens make an appearance around about 200,000 years ago. So in terms of the whole timescale of how long there's been life, how long there's been life on the planet, we don't come into it until right at the very end of this process. So if you take the whole evolutionary process, that whole sort of 4 billion years of life on Earth as a 24-hour clock, the mammals probably appear around about 5 to midnight. We make an appearance seconds before the clock ticks over to midnight. So, you know, it's a blink of an eye, the amount of time that we've actually been here on this planet. So yes, just to give you an idea of the slow changes and how things change incrementally, time over generation, until we get to this point where we're sort of highly evolved now. It's like massive, it's difficult to imagine and put into context the scale of time that we're actually looking at here. So we come to the question of the eye then. So I remember having a conversation with a colleague of mine a few years back about the evolutionary process, and he said to me, because he couldn't quite understand how the process could potentially work, and he said, how could something as perfect as the eye evolve out of nothing? And I thought, that's quite a bold statement for someone who can't see past arm's length amount of glasses. So perfect, very, very good, complex, amazing organism, amazing organ, but it's far from perfect. If it was, we'd all be doing something different. We wouldn't be sat here today, would we? So perfect is not a good term to use when we talk about evolutionary processes and things like that. But it is extremely complex. Now, I don't need to quiz everyone on the different parts. I'm pretty sure everyone's quite familiar with all those different parts. But when we look at the structure of the eye and the different functions, the different structures within the structure, it's massively complex, isn't it? There's so much going on in that one tiny little organ. And I'm sure we all sort of find it quite geeky and quite interesting ourselves, but it's very, very complex, it's very, very intricate. And there's a lot of different parts and different structures that interplay with each other. So it's hard to understand how something like this could actually form. But when we think of the difference between a complex organism and a non-complex organism, so let's think about a rock, for example. So a rock's not a complex organism. If you got the ingredients that make up a rock, shook them up, you'd probably get a rock. You could put those ingredients together in as many different orders you can think of, and you'd probably still get a rock of some sort. Whereas with a complex organism, actual living being, although the building blocks are the same, there's nothing different in the eye that's not found in other parts of the body in terms of down to like molecular structures. It is building blocks that are found in different organs in different parts of the body, but you couldn't just mix those up and get an eye. It has to be in that certain specific order. So it is extremely complex. It is hard to get our heads around that. But when we think about what we actually need for a visual system, is it that complex? Does it really need to be that complex just to have some kind of vision? So if we break it down into the basic components, we definitely need a photoreceptor. We need to be able to detect light. That's the bare minimum that we need. But what else do we actually need? Do we need a lens? Do we need an aperture? I'm sure everyone's done like pinhole cameras and things like that. Anyone that went through Abdo definitely did pinhole cameras. I'm sure we've done them at Optoms College as well, Optom Uni. You can get basic eye kits and make sort of basic pretend eyes. It's quite easy. So they don't have to be as complex as we think. They don't have to be as complex as what we have now to actually function. And the key thing to remember in this is that 1% vision is better than 0%. Anything that gives you that slight advantage over your competitors will enable you to have a higher chance of reproducing and passing those genes onto the next generation. So any kind of vision is better than no kind of vision at all. And it all starts with the photoreceptor. So we go back to the sun now. The sun is the source of all life, essentially, isn't it? So without the sun, we wouldn't have the energy. We wouldn't have had the planet in the first place. So the sun is what kicks off everything. But the sun, although it provides energy and it gives us life, etc., it's also quite damaging. It causes harm. We're well aware of, and we've just done a lecture about photochromic lenses, for example. So we're all well aware of the dangers of UV and the damage that the sun can do. So the first thing we ever needed was something to be able to detect when there's UV present in the atmosphere. And this all started through a protein called opsins. So opsins are little proteins that bind to vitamin A, and they're little coils, and the vitamin A sits inside the opsin coils. And then what happens is that the vitamin A itself senses the light, and it starts to change shape. And then this changes the shape of the opsin, and then the opsin sends a signal through the nervous system to say that light's been detected. And this was thought to have first occurred around about 600 million years ago. So we're going way before complex organisms. We're going right back to single cell organisms and things like that. And it's related to what's called oxidative stress. So we know there's damage in UV. We need something to tell us that there's damage in UV so that we can respond accordingly. Whether that's moving out of the UV, whether we need to be aware that there's UV so we can move into an area where there is light, if we need light, in terms of influencing our circadian rhythms, etc., and all that kind of stuff. So just being able to detect UV is the first step in forming an eye. The next part we need then, so we've got these opsins, we can detect when there is UV. But the next thing we need to know is where that light is actually coming from, and that's the next big step. And this is where we had our next sort of evolutionary stage, which was dark pigment cells, which shield the photoreceptor cells from one side. And once you have that dark surface, basically a choroid, behind those photoreceptor cells, we can now detect which direction that light is coming from. So we've gone from just being aware of light to now being able to tell the direction that light is coming from. So again, if we need to move into or out of that light, we've got that option there. And we still see this in nature today. So this is called a Euglena, and this is a little sort of single cell organism that's found in water. It's like a little flagellate, it's got a little tail, and it swims around in the water. And what we have is an eye spot. So that little red dot there that you can see, that's what's known as an eye spot. And it's literally a layer of photoreceptor cells with a bit of dark pigmented layer behind it, some dark pigmented cells, and that's how it navigates around in its environment. Just basic photoreceptor cells, pigment cells, and it can tell where the light's coming from, whether it needs to move in or move out of the light so it can find food or it can find shelter, etc. So we've got our first almost functioning eye in that sense. So the next step then, so once we've got that direction determined, we need to form like a crude image. So we need to be able to form shapes and sort of find out a bit more about our environment. And this happens with an eye cup. So that dark layer of pigmented cells, again through a slight random mutation, some became slightly more depressed than others, and that formed into like an eye cup. And this was our first step towards low resolution vision. So what we have with an eye cup, we have a series of photoreceptive cells that are all pointing in different directions. So we've got the directional aspect where we can tell where light's coming from, but we can also start to form crude images, build up an idea of shapes and what's actually in our environment, distances, and it's the start of sort of 3D vision basically, which obviously again is much more advantageous than a simple eye spot. And we see this again in nature. So this is a planarian flatworm, and you can see just there its little eye cups. So it looks like it's got looks a little bit cross-eyed, doesn't it? But those are basic eye cups, and we still get flatworms today. They're still found in water and in different types of environments and stuff like that. But the way their eye cups work is you've got the PHC. Those are the photoreceptive cells, and then you can see that dark ring around it, that depressed cup, which is the pigmented cells, and that gives it its basic crude vision. It doesn't need particularly sharp vision. As long as it can navigate around, as long as it can detect where it is, then it can thrive in its environment. So the next question is then, how do we improve on that? And we go to the pinhole camera. So this is called a Nautilus, and this is related to octopuses, which I think is the plural for octopus, isn't it? So we'll go with that anyway, if it's not. So the Nautilus has a basic pinhole camera for an eye. And what we see is that we have that depressed eye cup, but we also have a continuation of tissue enveloping around it, which gives us this sort of pinhole, gives us an aperture. And then because this is a sea creature, that depressed area then fills up with water, and we've got ourselves like an aqueous humour. We've got a vitreous basically. So water fills it, and that provides a different kind of refractive element as well. And then through controlling the size of that aperture, they can again adjust their focus and adjust how much light is coming into and hitting the retina before it goes off down the optic nerve. And again, another step that's provided it much better chances of survival. It's advantageous. It gives it that extra bit of possibility of survival, gives it the ability to hunt better, etc., evade predators better, and all that kind of stuff. So the drawback of this kind of eye, though, is that it only really works in water. Because if you take that Nautilus out, then it doesn't have that option for that depression to fill up with water to form that kind of aqueous humour. Another potential issue is that you're going to get a lot of bacteria, you're going to get a lot of things sort of enter into that area, which could then cause problems like infections and stuff like that if it's not regularly sort of cleaned out. So the next step then is to protect that retina from those kind of elements, from those kind of invading bacteria and stuff like that. And this is where we get the cornea. So we want to avoid those parasites and bacteria coming through the aperture. So again, we have a continuation of that tissue layer, and there's different. Again, if you go look throughout the animal kingdom, there's lots of different examples of cornea and what they're made out of. Some are made out of similar materials to things like claws and nails and teeth and things like that. Whereas ours, we're looking at very similar tissue. They share an embryonic origin as the skin. So we're talking about the epithelial cells and things like that. So it's not a new material that's suddenly evolved. It's a continuation of what's already there. It's continued to grow over that aperture and enclose and protect that internal structure. And again, this is what sort of allowed us to form that aqueous humour, which then allowed us to sort of, in terms of like circulatory fluid, oxygen, waste, nutrients, and transport that around the internal structure as well. So we're pretty much there, but there's still another stage that we could go through. So the next stage, which is the game changer, is the crystalline lens. And this allowed us to, this is what really sort of defines the type of eye we have. So we have what's known as a camera eye. And it's the crystalline lens that really sort of levels up. Sorry to use a levelling up phrase, and I sound like Boris Johnson there, but it kind of levelled up the eye there, didn't it, and give us that ability to focus in different focal lengths, to have that pinpoint sharp vision, and to be able to sort of get that better, high contrast, high quality vision in that sense. Now, the bulk of the lens itself is made from different types of crystalline. Again, crystallines are found throughout the body in different organisms, different organs in different parts of the body as well. So then, again, they're not unique to the eye. And they're formed mainly from what's known as alpha and beta gamma crystallines. And these are called heat shock proteins. And the job of a heat shock protein is to basically alert a cell to some kind of change in environments, whether it's heat, whether it's changes in temperature, UV light, where they form a role in sort of the healing of wounds and things like that. And it's these heat shock proteins that respond to these stressful situations and enable us to send signals down the nervous system and things like that. And that's basically how we formed our crystalline lens. So there's 5 different types of heat shock proteins in total, and their job is, like I say, cellular homeostasis. It's to have that stability. They can detect changes, they can detect stressful environments, and then the body can react accordingly to them. Okay. So there we have, like I say, we're pretty much there in terms of the structure of the eye. There's lots more extra functions that we have as well. Obviously we've got eyelids, we've got ocular muscles and all that kind of stuff, but we've pretty much got a functioning eye that's going to give us a big advantage over competitors in the natural world. So the question is then, how long did this process take? So how long did it take to go from that basic eye spot to the eye cup to a functioning camera eye like we have today? Any guesses?
365,000 generations.
Someone was paying attention to the quizzes we had. Yeah, so it's not as long as you think actually. So there's a study done by Nielsen and Palja, and they worked it out to approximately 364,000 generations, which is roughly 500,000 years. So when we think about, we put that into context of that 4.5 billion years that we've had life on this planet, it's quite a small period of time, isn't it, for that to develop something so complex, just by a change in each generation and the passing on of those genes generation to generation. Okay. But the eye itself, it's not the whole package, is it? It's part of a visual system. And for us to form and interpret the signals that the eye sends to us, we need to have a brain, we need to have a nervous system. So we have those electrical impulses that are triggered by the photons, the light stimulating the retina and sending those messages down the nervous system. But without our brain, we can't really determine what that image is and, and the detail behind it. So, as complex as the eye is and as fascinating as it is, it's not everything we have. We need a brain for it to work to the potential that it does today for us. And again, that helps not only with vision, but also our circadian rhythms, which is very important for us, in terms of rest and recuperation and stuff like that as well. But there are some animals out there that do have complex eyes but don't have a brain. So this is the Cladinema radiatum, which is a type of jellyfish. It sounds a little bit like a Harry Potter spell, but it has a complex eye that's very, very similar to ours. So you can see the eye pointing at my screen, but you can't see it. But I'll point it there. So you've got like this bit here. That's the eye. So it's not the same. It's not the same shape as ours. It doesn't look like ours, but it's a camera eye. So it works the same, basically the same way. Except these jellyfish don't have a brain. So instead of them interpreting it as an image and forming an image like we do, it just passes on those signals directly to the muscles, and then the muscles respond, and then the jellyfish can navigate throughout its environment. But the reason we sort of brought in this particular jellyfish is it's been quite instrumental in a lot of research about what's known as master genes. So the master genes are almost like a conductor in an orchestra. So obviously genes play a huge role in shaping us, in determining how we develop, how we evolve, etc. But there are certain types of genes called master genes that basically work in collaboration with other genes and kind of orchestrate them and conduct them and trigger them to work in a certain way. So they basically, they kind of control things. They're like the gene foreman, if you will, the conductor. And like I said, this was first sort of discovered by Basel University, and their research found that all animal eyes share a common origin and a common master gene. And although the details might be slightly different, it all comes back to this one master gene, which is known as PAX6. So if we think about the process of growing an eye, it's definitely complicated and involves a lot of interplay between different gene groups and different types of genes to give us what we have. But it is this PAX6 gene that basically causes all that to happen. That's what actually controls everything. It's like the CPU, basically, in that sense. And the PAX6 gene then, so the gene that causes all this, was first discovered in 1994 by a guy called Walter Goering. And if there's faulty copies of this gene that are passed through, then it can cause sort of serious conditions. It can cause serious eye problems for different animals. And they've noticed these in animals as diverse as flies and rodents. And you can also activate the gene in the wrong part of the body, and you can produce eyes where they really shouldn't exist. So, for example, if we have a look at the eye of a fly. So a fly it doesn't have a camera eye, it has a compound eye. And a compound eye is basically thousands of tiny little eyes. And what they did was they got this PAX6 gene, they triggered it on a different part of the fly, on the fly's leg, and they were actually able to grow an eye on the fly's leg. So that little red bit there, that's the fly's eye. And you can even take these PAX6 genes from different animals and you can put it onto another different animal and it will still be able to trigger the development of an eye. So again, they've done this with a PAX6 master gene from a mouse, implanted it onto a fly, and they're able to grow a fly's eye on a fly in a different part of the fly's body. So it's mind-blowing really, isn't it? It's just really interesting that you can do something as crazy as that. But like I say, it's that PAX6 gene, that's the root of it all. It's that controlling master gene that triggers the other genes to kick this process off.